Today, I'm very honored to welcome someone I have had the privilege of knowing for over 15 years, Dr. Mahendra Rao. Mahendra and I first connected through our shared passion for the potential of iPSC or induced pluripotent stem cells, particularly the promise of hypoimmune iPSC-derived therapies to create scalable allogeneic solutions that can serve all people, not just a few. Mahendra is a true pioneer in the field of stem cell biology and had a remarkable career spanning academia, industry, and government. He was the founding director of the NIH Center for Regenerative Medicine. He's also served as vice president of regenerative medicine at Life Technologies and has been a key advisor to global initiatives shaping the future of cell therapies. Today, he's continuing to drive innovation as the founder and CEO of Implant Therapeutics and CSO of Vita Therapeutics, where he's pioneering work in hypoimmune cell therapies. What I have always admired about Mahendra is not just his scientific brilliance, but also his commitment to really translating science into accessible real-world impact. I am so excited to have this conversation with him today and to share his insights with all of you. Mahendra, welcome and thank you for joining me.
Good to see you, Ruby.
You too. So I'm going to start by really talking with you about your career and your vision. And one of the things that really made impression on me is that you were the founding director of the NIH Center for Regenerative Medicine. And you were at the center of policy and translational science. So where were some of the most impactful lessons that you learned in that role?
So, there are many different choices you make in your career, and they cause big changes in what you do and the direction you take. And sometimes there are reasons for those choices. So in my case, the biggest change occurred when I was training as an MD physician in neurology, and I wanted to do research work. And then I had a choice between doing research in clinical trials or doing research in basic science. And that was a really big change in my career because I opted to do research in basic science and decided that because of my training, I needed a PhD program. And so I joined a PhD program, which changed the direction of my career. But joining the NIH was another pivotal moment. And it was, as you pointed out, I'd always wanted to make a difference. And the government is the largest sponsor of research, and at that time had made a huge effort to try to lead the direction of this new field and make sure that there were appropriate regulations in place to make sure that the field could move forward rapidly. And in addition, be able to provide appropriate funding and grants. And so it was really exciting to be at the forefront of that change that was occurring. And so that was a big change in my career, where I gave up working in a commercial setting to work as a government employee. But I don't regret it at all. And it was really a wonderful way to be able to make a difference.
Yeah, no, definitely. And the cell lines that you made, the GMP grade iPSC cell line, it's being widely distributed and used by a lot of groups. So that's one of the, biggest thing that you actually created while you were at this role, and really is now benefiting a lot of groups who is developing iPSC derived cell therapies. And I thank you for that. And so looking back, is there a moment, I guess you kind of mentioned it, is there a specific moment in your career that you consider a turning point?
The biggest turning point came when I was first working for the government, and there had been this discovery of embryonic stem cells, right, made from fertilized embryos. And there was a lot of excitement in the field because for the first time we had identified a pluripotent population of cells that could be maintained in culture. And then, as you know, it got mired in both legal and ethical issues in being able to move that field forward. And I really feel that one big turning point was this ability to develop adult derived pluripotent cells, which was where the Nobel Prize was given to Dr. Shinya Yamanaka for that discovery, because it took a field and bypassed the ethical issues that were there simply because of the source material that was used. And so that made it really possible for everybody worldwide to be able to look at working on these cells. And so that to me was a big turning point, because I was reflecting at that time on what would I do if this was against the law. So,
Wow. So yeah. So that actually, I shared exactly the same comment about my turning point. Is exactly the same thing. So I was also working at Stanford University on embryonic stem cells. And then 2006, Yamanaka discovered iPSC in the mouse first, and 2007 in the human. So that's when I actually got really excited about the potential of iPSC. And then a few months later, I started Applied Stem Cell. So that's my career turning point, is really started Applied Stem Cell because of iPSC. So I guess it's no surprise that we kind of hit it off, because we share a very common vision and passion about working with iPSC. And this is the first time I asked you this question. I'm just so happy that that we actually came, to a very similar conclusion, inspiring discovery of iPSC. And so let's talk about scientific innovation and how to translate science into real life impact and applications. So, so the one of the things that we've been always talking about, particularly in iPSC derived cell therapy, there are several bottlenecks, of course, and one of the biggest one is for me is really immunogenicity. So quite a few years ago, you and I talked about how we can make a hypoimmunogenic iPSC. Right, so that was a while ago, and now actually there are hypoimmune cell lines available, and some companies are using them, and they got some good results, right? So my question to you is, different companies or different groups have different strategies to edit the cells to make the cells hypoimmunogenic. So my question to you is, what are the key gene edits in your point that would make iPSC hypoimmune, and how do they work mechanistically to evade immune detection?
it's a complicated question. And now, given your immunological background, you'll be well aware of that. So let me try to simplify it a little bit before I answer the question in detail. So, as you know, your body has a system for recognizing self from non self, right? And anytime there's some kind of non self cell type or an invasion of a living organism, then your body activates its immune system to be able to destroy that invading organism or the transplanted cells or the invading cells to be able to do it. And it uses the same immune system to try to protect you from cancer, because cancer cells sort of lose their identity in some fashion, and the immune system has evolved to be able to take care of that as well. And so scientists, being innovative, looked at all of this and said the best way to be able to bypass the immune system when you do stem cell work is to be able to co-opt the same natural systems that other organisms and cancer cells have used or we have used when we do organ transplants. And so I can divide those into three different types of ways that people have done this work. One is what we call immune suppression. So you can imagine that if you take the immune cells and just pretty much destroy them, then you would be able to transplant cells. Of course, you'll have to keep the immune system dead for a reasonably long period of time, and you have to worry about all the consequences of that. But that was the first successful method of being able to do it, and that's how bone marrow transplant succeeded in the first place, which was the first kind of stem cell transplant that was done. And a second way that was done was to do what was called immune evasion. So don't destroy the immune system, but fool the immune system, right? And to fool the immune system, you had to bypass all the signals that the immune system uses to recognize self from non-self. So the first most important thing is we have what are called HLA class 1 and class 2 genes, which are used to be able to detect antigens which are present uniquely on yourself and which are different in every human being. Even among twins, there might be small differences in transplant antigens. And so, if we could bypass that, then we would be able to transplant cells and be able to evade the immune system, which would not recognize these cells as being foreign. And so, first simple way that they could do this was to just knock out the immune recognition system. So, knock out the class 1 recognition system, knock out the class 2 presenting system to be able to do that. And gene editing techniques allowed us to do that in an easy way. So that was the first step in doing it. Of course, it's not enough because the immune system is not that simple. And so there's a second part of evading the immune system that had been recognized, and that was the innate immune system, which recognizes things based on slightly different parameters. And NK cells in particular recognize whether the cells have an immune signaling mechanism or not. And so if you knock out the immune identity system, then NK cells will be activated. So scientists then provided a way in which they could bypass the NK cells as well. So there are molecules which are known which can suppress the NK cell system. And then that was one way that one can add it to the class 1, class 2 null mechanism to be able to do that. A third important component of the immune system is what are called macrophages. And macrophages are the first line defense. They actually look at something that's foreign or which is causing damage or inflammation or hurting the body, and then they go in and they kill the cells, and then they eat them. And then they take these antigens and activate the T cells and the B cells and the NK cells by presenting the antigen as well. So you need some way to be able to bypass the macrophages as well, because then you could stop the immune system activation right at the beginning. And what people discovered was that there were certain signals that were present on normal cells, which were called don't eat me signals, which allowed the macrophage to ignore a cell, even if it had an aberrant or a different class 1 or class 2 antigen epitope on it. And so people figured out that they needed to overexpress a don't eat me signal to prevent the macrophages from acting on the cells. The last piece that people had to worry about was the complement system, which is an early system which takes advantage of pre-existing antibodies which are present to antigens that you've been exposed to, which are non-self. And these exist even when you have to do a transplant. And so you needed some method of being able to bypass the complement system as well. So surprisingly, people found that the body adapts. And so you could just do a subset of all of these changes. So initially, people started with looking at an entire subset of changes. So they tried to bypass the NK cells by using inhibitory HLA antigen or being able to use a different component of to fool the NK cells. People knocked out class 1 and class 2. People expressed CD200, which is an inhibitor of T cells, etc., to be able to do that. Or they combined it with using small molecule inhibitors, which tamp down the immune system. And they added a complement and a don't eat me signal. But Sana was one of the early companies which then showed that maybe it's as simple as just expressing a don't eat me signal at high levels. So they use CD47 and just knocking out class 1 and class 2 to bypass the main component of the immune system, the T cells and the B cells, to be able to get that done. And that turned out to be enough that the NK cells themselves also did not attack. And as you know, it's gone into clinical trials, and Sana has been treating patients for diabetes using islet cells derived from hypoimmune cells to be able to do it. Other groups have tried slightly other strategies, but that's the basic idea.
Right. Yeah. Thank you so much for going through this very complicated pathways and processes. And yeah, so I totally agree. It's just immune system is so complicated, and but whatever genes that we modify, and we're a step closer, right, to what so-called universal donor cells, right? So and some the sauna the data is very encouraging, so it's actually pretty straightforward to knock out class 1 class 2 knockouts plus CD47 expression was actually showed pretty good immunoprotection in their study. So that's very encouraging. And then, if we could engineer some of the other pathways, as you mentioned, uh, um, they're also like dendritic cells, for example, and then also just a very general apoptosis pathway, right? So if things can be um, um, changed or modified there, and then we'll get better cells as donor cells. They're immuno evasive. So yeah, so this field is evolving. That's what I see. But as we see that all these manipulation or putting in the genes or knocking out the genes would require gene editing technology. So to some extent, when we talk about hypoimmune iPSCs, we cannot do without using particular gene editing technologies. So you and I, we talked a lot in the past about gene editing technology, and maybe you can really kind of give us some idea, because you also talked about you've worked with some more advanced CRISPR technology. CRISPR-Cas9, everybody knows it's a revolutionary technology, but also it comes with a lot of limitations too. So maybe you can talk about how what are the technologies that you think would be very useful in making these hypoimmune iPSCs?
So, Ruby, as you know, we've talked a lot about all of this, and you know that the one big problem always is that when you're manufacturing cells, not only do you have to worry about the technology and what gets done, but also freedom to operate, right? So that you can actually do it and it's not a monopoly of some company and you're able to get these licenses to be able to get that done. And I know you at Applied Stem Cell have made a big effort in being able to get that done, but it's something which has been a roadblock. And so, one thing we've been looking at is which technology will work to be able to knock out the genes that we need to in class 1 and class 2, as well as introduce and overexpress the candidate genes like the don't eat me signal or everything else that can be done. And so I guess I'm revealing my age and how long I've been in this field. We started trying to do all of this work with zinc fingers, and then we followed it up with talons. And they all worked, but the efficiency wasn't that great. And then with CRISPR Cas9, it became really exciting because one could do this at a much higher efficiency, even though there were many patent issues. And you probably heard about all the lawsuits that have been going on between different companies and between the uh, Boston group and the Berkeley group in terms of patents and etc. But one thing that's happened is that it's become clear that CRISPR-Cas9 is a thing that's been used by many, many bacterial and plant organisms to be able to protect themselves from disease. And therefore, there are many variants of the basic technology that was identified by the groups earlier. And so there's a Cas12 system, which is a little bit orthogonal to the Cas9 system that's available. And you know that you've used MAT7, which was one of the earlier enzymes that was discovered in this pathway. But now there are over 1,700 different CRISPR Cas9 systems that have been identified. But the biggest excitement has been this whole thing that we can now engineer them because we understand enough about protein folding and protein design and protein activity so that we can make completely new synthetic CRISPRs as well. And as you know, there was a paper last year on this open Ep.1, which was a completely novel protein designed based on the principles that we understood from what has been done with CRISPR systems in being able to do that. And so we found that those really work. The other exciting thing that's happened with CRISPR has been that you have CRISPR systems which don't just edit DNA, but can be used to activate or methylate genes to start and shut them off, and also which look or focus or direct themselves at mitochondria or RNA. So the Cas13 systems work on RNA. So you can see the whole field and the potential of how we make knockouts and whether we need to make them regulatable or inducible knockouts has all become much more feasible with all the new advances that have worked with CRISPR. In, in our hands, we have found that there are many, many CRISPRs which have allowed us to do this kind of editing that we do, which, to be honest, is on the simpler side for immune evasion compared to the more precise gene editing and repair that you have to do for point mutations or for precision medicine. to be able to do that.
Yeah. Yeah, so I agree. It's just like the technology development. It just evolves so fast. There's so many different alternative tools out there. But one of the biggest concern for therapeutic application is the IP landscape, because a lot of those CRISPR-Cas technologies are kind of subject to the patent protection for CRISPR-Cas9 system because they are the foundational technology, right? So because the components um, and all protected in the patent, and then the litigation between UC Berkeley and MIT is just being ongoing for a long time now and still ongoing, and who knows when it's going to end. So that it's unfortunate that that's happening, right? It's really monopoly, actually not letting this amazing technology to be widely used. So but what we have at Applied Stem Cell, as you mentioned, is that we were very fortunate, and also in Scripta at the very beginning, they were very encouraging that people to use their technology with very friendly licensing terms. So we were able to get MES7 license to use that to do knockout, and then we have our own target serum recombinase based technology to do gene insertion. And the technology that we use target is actually originally invented at Stanford University. And our head of R&D, Alfonso Farruggio, and myself are co-inventors of this technology. And we have to continuously develop this technology at Applied Stem Cell. And now we have next generation technology called S-Select. It's even better than the old target technology. So I think that these technology platform would help us to make the ideal cell line as donor cells to make cell therapy product a much cost-effective process, and make it more of one product for many patients, and allogeneic is the way to go, right? So,
So I
um,
absolutely agree with you, Ruby. And I want to emphasize one thing, and I'm not just being polite, but when I have to choose a CDMO or somebody to work with, you need to have somebody who has rights to a particular technology so that they can use it. And as you pointed out, Applied Stem Cell has been able to do that. But there's a second really important component that I found missing in many of these other companies, and that's the technical expertise as well. And again, there is what I found really attractive about Applied Stem Cell has been that you are the inventors of the technology. So you own the technology, so you have the expertise with it as well. But that's why we've been so happy working with your team, right, in being able to get stuff done, because we know that we won't have a problem with having to do a lot of technology transfer. But that's how we look at many of the CDMOs. And I think that's a real big strength that now we have companies and CDMOs which are trying to make sure that they have access to this kind of technology so that the end user has a seamless experience. And it's good to see that Applied Stem Cell has been a leader there with this new technology that you pointed about S-Select as well. So.
yeah, thank you so much, Mahendra. I really appreciate. Yeah, so yeah, that's very important. The technology platform, the expertise as a CDMO, I think it would really benefit the drug developers and therapeutic companies. So yeah, my next question is, I always thinking about it is, we're working on these hypoimmune cells, and hopefully that would solve all the problem, right? That's my wishful thinking. But I still think I actually, there are other, the current immune suppression, would that be still necessary, or would customization remain necessary? So that's something that I'm thinking about. What are your thoughts? I just kind of want to hear from you.
So, I'll give you an answer which is one which is theoretical, and then the other on what we know so far, because we don't have enough data to be able to get a clear answer. So, on a theoretical side, and based on what's been done with bone marrow transplants, where we have almost 50 years of experience, is that there is something called acute rejection and something called chronic rejection. And in the acute rejection, we found that this class 1, class 2 null, and being able to do don't eat me signals can all work quite well. Or we can have antibodies which will work. The longer term chronic rejection depends on a slightly different set of pathways. And people have found that people who have accepted bone marrow transplants can still reject after 10 years, 15 years, sometimes 20 years, right, after a transplant. And that depends on how the immune system changes in those 20 years. And people have identified the components, and there are minor blood group antigens, there are minor other kinds of antigens, and they are all tissue and cell type specific. So my feeling is that yes, we've solved the first big major barrier, but there will be some kind of personalization and tweaking that will be necessary, and will depend on people. Just like when you have autoimmune disease, some people get it and some people don't. Some people are more sensitive and some people are not. And so we will have a baseline therapy, but we will have to probably tweak as we move forward when we're looking at transplants.
Yeah, I agree. I agree. And I guess it's a little bit too early to really understand. So I guess we'll see. Um, so let's move on to the regulatory policy and global landscape. So I mentioned earlier that you were, when you were at the NIH, and you headed the effort of making the first GMP iPSC line. And that particular iPSC line had a drug master file with the FDA. And so since then, I think there's several drug master file has been done for iPSC, including applied stem cell. We have a DMF on our iPSCs. So what is your perspective on the value of drug master file for iPSC cell bank in terms of accelerating cell therapy development?
Well, I think it's a huge benefit, and that was the reason why I insisted when we started that we would go to the FDA and at least get the whole process in place. And I modeled it after what had been done with all the producer lines which are used for production of viruses or proteins or biologicals, where there was this precedent that if you were making a product as a starting material, then you could get a drug master file for a living equivalent of being able to do that. And I would say that nobody, if they have a choice available of having a drug master file and a characterized line, they would opt for anything else. So I think it's an absolute necessity. And that's been the reason why the NIH line, which was made initially, has been used by such a large number of companies, because they had that comfort that the FDA would not reject it. And since it's starting material, you know, it means that anything that happened later, if there was a question on your starting material, then you would have to start from scratch again, right? It's not a downstream problem. And so I really personally think that no CDMO can really try to sell iPSC lines without having a drug master file available for it. I was really hoping when I first did this that there would be reciprocity between the different regulatory agencies so that a drug master file with the FDA would be used and accepted internationally. But that's not the case. And no other regulatory domain has the same drug master file equivalent program that we can use, which has been a little bit unfortunate. But at least it's a start, and you can use the whole set of copies that are there and present the set of documents, and we found that they're largely similar, but they won't just accept the drug master file citation.
Yeah,
These are the
that is
requirements.
really, yeah, too bad. I guess it would. I don't know when that's going to happen because every country and territory have their own requirements.
Yeah.
Yeah.
But at least what has happened is that it's become the next target for CDMOs, which are global. Is they try to get regulatory approval for the same cell line based on the documentation that they've collected for the FDA uh, in Japan and in Europe, in the EU, and that's become possible. And so you can at least have that kind of universality with your cell line. And
I
that's been
see.
the target for a lot of the CDMOs now.
Yeah, yeah, I see. Yeah. So yeah, so for us, because the drug master file is almost like you the part that you would write for R&D package under CMC. So everything has to be documented, and the donor's eligibility screen and all the documentation, the consent has to be in. So and batch record has to be in. So it's basically the CMC part is pretty much written, and all you need to do is just to cite the drug master file number. I think it's a huge benefit to drug developers who want to use these cell lines. And I totally agree with you. Like, whoever developed these starting cell lines should have a drug master file with FDA. Otherwise, I just don't think it's even meaningful, right? When you claim you have a GMP iPSC line, if it's not documented with the FDA, it really doesn't speak very well about your cell line.
That's
So
right.
yeah, so speaking about, talking about different regulatory countries and territories, and so you have worked with international agencies and governments, and so I think it's similar thing is like, what kind of role should public institutions play, or just what we can do in accelerating this field, kind of at least kind of unite across the different agencies and the government regulations, so that we can actually move things faster across countries.
Thanks for asking me a leading question, because this has been my pet peeve on how we can accelerate this and make it sort of globally available. And I think that there are two really important things that public institutions can do. One of them is something that has been done in the digital field, uh, and been done with computer scientists, and that's what's called this common use patents, right? And so there's something called FRAND rules, or fair, reasonable, and non discriminatory rules for patent licenses. So you can't use your patent as a monopoly to be able to use that. And you have what are called key patents, and those have to be licensed even to your competitors. And the rules are that it's equitable, right, in terms of the licensing fee, so that it's done. So those FRAND rules have been a really common thing that all the software developers have gone through and been able to get sort of agreement on. So even though you're competing, you're sharing, and that has allowed the field to move forward really rapidly. We don't have that equivalent, even though a lot of people have tried to be able to get that done, and that's not happened. The other thing for which there's been a lot of effort, but and luckily the United States has taken a leadership role in that, and that is harmonization of regulations. And there have been a lot of ways that the FDA has tried to do this. There have been standing committees that they've set up which are international, and they actually do cross training, which has been really important in being able to get that. So the Korean FDA sends scientists to the US FDA. Japanese FDA PDMA scientists have come to the FDA, and vice versa. And they have inspection rules for sites which are in different places. And that really can't be done by any company, right? Or it's just the organizations or self regulation like FACT, which does that for CDMOs, right? And having training programs. So that is what you need, a central government or public authority to be able to do that. The last piece, which I find is really critical, is training, standardization. So you can make regulations, just like you can see now that we have USB-C for phones, everybody can use the same charger, right? If we had some kind of standardized tools or tests that are available, then manufacturing would be much easier, and that can only be done by regulators. And so I think that's a really critical role that public organizations can.
Yeah, I think it is very important. This is, I think we all need to work together and really try to make this happen someday, right? And get
Yes.
closer to our goal. Um, yeah, so very good. So I cannot, not mention this new the headline in the last couple of weeks about this story, baby KJ. Um,
Yes.
so basically, so baby KJ received a custom on-demand CRISPR therapy, right? And now he's home. He's cured using CRISPR editing. And so this work was really a collaboration of many teams. So UC Berkeley, their IGI, their institution, and it's headed by Jennifer Donna and Fyodor Ernov, and partner with CHOP and Penn, as well as Danaher, and some other, I think, several other companies. So it was really a very big team effort to make this one case a success story. And they were able to secure an FDA R&D within weeks and achieved, successful base editing in vivo. Um, so I I was very inspired by the story. Mainly because, of course, this is very exciting. Um, and then another thing is this model that Fyodor Ernov mentioned. I heard about this model about a year ago when he talked about this at the ISCT meeting. Is really this open IND um, idea. So basically, you set up a platform technology, and then the things that change is like a plug play kind of thing, right? So if we could do something like that for hypoimmune iPSC based cell therapy, right? So imagine that we start with the same hypoimmune iPSC lines that's validated in a clinic. Of course, has to be tried in clinical trials, right? And validated and proved to be safe and efficacious. And then this iPSC line can be used as platform technology. And then the additional things just by adding different genes, right? So I like to pick your brain and really, hear your thoughts about, are we, can we actually do that? If so, how can we make this happen?
So, you made a very important point, and it's something that, luckily for us, the FDA has been quite foresighted in being able to think about this. So it all started when we had this siRNA based technology, which was first used in patients. So what people realized there was that you were synthesizing the same oligo roughly about the same length, and you were delivering it the same way if you were doing it in the CNS, and the doses were roughly about the same, and the mechanism of action was the same. So they proposed this idea of being able to do this where the IND would be very straightforward, where you just say I'm changing this for this different indication, but my monitoring, my readouts, and everything else will be easy. And to prove that point, the former founder of the company set up a nonprofit, and they were able to show that a small company, which has got just 5 people as a nonprofit, could submit 40 different INDs, right? So the paperwork was virtually identical. They had to do it, and they could vary it along the model of an investigator initiated trial. And then what you saw with what happened with Berkeley was exactly the same thing, right, in being able to do it. So the FDA looked at this and they made uh, the following statement, and that was Peter Marks. And so I have to give him credit for having sort of pushed the idea. And the idea was we can define something as platform technology. A company can come to us and say, this is a platform technology. And an example of one such platform technology, which they approved recently, was AAV, ethno-associated virus. It's used by many different groups, but it's the same capsid. It's only the gene of interest which is changed. And so the idea was we can designate this capsid and this process of manufacture as a platform so that for every indication you can do the same platform approach. And the hope is that we can take that one step forward along with that for exactly what you pointed out with iPSC, to have iPSC which are gene edited designated as a platform. So one has to go forward and have that designated as a platform technology, and then one could be able to do exactly as you proposed. And I think that's going to be a very important piece. And luckily, the regulators seem to be on the same page to be able to do that. And they have one sort of antecedent for that, and that is cord blood. So, cord blood was declared as a sort of platform technology for transplants, right? And so different banks could make it and manufacture it differently. But since it was a platform, they all had to just show that the dose was the same per kg body weight with different release criteria and different manufacturing as a sort of platform concept for treating multiple different diseases. So my hope is that that's exactly what will happen with iPSC.
Yeah, so that sounds really encouraging, and also to hear that FDA is open to that. I have watched recent FDA CBER um, roundtables and all that. I think they're very amenable to these novel ideas, and also really because it's saving lives here, right? So you cannot take 10 years to develop a drug anymore, because these especially children, pediatric indications, and so these um, like CRISPR or gene editing technology or iPSC technology could actually potentially save lives, and as an on-demand product. So I just
So,
feel very, I'm excited, and I feel very fortunate to be able to work in this field, and for a while, I guess you have the same experience, and now we're actually seeing the real application is happening. So
so
that's
Ruby,
very
uh,
encouraging.
I know you must have been thinking about this for a long time, because that's the other part of your technology which really has excited me. And that's this whole thing about target and using it in a defined site. Because think about it, it's exactly the same, right? You're using the same targeting molecule, you're using the same enzyme, you're using the same locus, the same procedure for electroporation. So it's a platform. It's only the gene of interest that changes, right? And even the silencing mechanism or expression mechanism remains the same. So that could be designated as a platform technology, and it would save so much time and effort, right?
Yeah.
You're using the same cells with the same engineering to deliver hundreds of different products, right? To be able to
Yeah.
do that.
Yeah,
So,
absolutely. Yeah, absolutely correct. So, FDA has this new designation called Advanced Manufacturing Technology. So that's the type that would be a good fit for us. Basically, it's the same iPSC line, the same target technology, and all the reagents are the same. The process the same. The only thing that changes is the gene to be inserted. So yeah, so we're looking for partners that who can work with us, because we do need to have clinical data in order to apply for such a designation, and that would definitely benefit later on a lot of product development, right? Once this designation is obtained. So
I think if you advertise it a little bit more, you will have many people very interested, because, the advantages of getting that are so obvious that they're really nice.
yeah, let's hope people will hear our podcast, and this is a call for partnership here to
No, but it'll be very exciting, and the
yeah.
technology looks really good. And just as you know, whenever something is really good, there's always competition. So I've heard that there are a couple new companies trying to improve on the technology that you provided, and you also said that you have a newer method of being able to do it.
Yeah,
So
yeah,
a plus.
yeah. I know a good technology. I think competition is good. That's what makes science more exciting, right? So
Yes,
every day there's
accelerates.
something new. Yeah. All right, very good. So I really appreciate your time, Mahendra, and your insight. It's always such a pleasure talking to you. I don't know if you remember, I always said I learn so much by talking to you every time. So this is not exception either, right? So this is always such a learning learning process for me too. So I cannot thank you more for your time.
Thanks, Ruby, and I look forward to seeing success.
All right. Thank you.